through the vasculature. And so cells escape, they enter the bloodstream, they often move towards stiffer tissues like the bone. And in the case of ovarian cancer, these cells grow in the peritoneal cavity, in the ovary or in the fallopian tubes, and the cells are sloughed off and they stick to things in the peritoneal cavity. And so they're moving in a softer environment, and they're metastasizing to soft tissues, like the omentum, which is the fat pad within the peritoneal cavity. And so there's differences in the method that the cells undergo migration by, and there's also differences in the tissues that they metastasize to. And so we thought that this might cause some differences in the way they interact with surfaces of different rigidity. And so we can use polyacrylamide gels to generate these small surfaces that we can seed cells on, and we can generate them with elastic moduli between 100 and about 150, well, between 100 pascals and 150 kilopascals, which is a very wide range. Most tissues are between 1 and 50 kilopascals, other than heart bone. But a lot of our soft tissues, and the tissues that I'm interested in, are in this range of between 1 or even 10 and 50 kilopascals. And so for our studies, this is an excellent model. We have high resolution with traction force microscopy within this rigidity range, and so it works out nicely. And so we use several different cancer cell models for these studies. We use 2 metastatic cancer cell lines, including MDA MB231. If you do any work with cancer models, you probably have heard of MDA MB231. It's a very common metastatic breast cancer cell model. And then SCA3, which is a metastatic ovarian cancer cell line. This is a serous ovarian cancer, which is one of the most metastatic types of ovarian cancer. And so it is a highly invasive type of ovarian cancer cell. And then we use cells that were thought to be non-metastatic. We always have a reference cell in any study we do. And so with our stem cell studies earlier, our reference cell was a fibroblast. And we had human fibroblasts in our human studies. And here we have non-metastatic cancer cells, which represent a less aggressive cells that we can compare our force profiles to. And so we use several non-metastatic breast cancer cell lines, MCF7, MDA, MB361, and then we use one non-metastatic ovarian cancer cell. We use 2 rigidities for our polyacrylamide substrates, including 3 and 35 kPa. This is similar to what we might expect for fat, and this is similar to what we might expect for a more rigid tumor. And then we use this kind of group of types of experiments where we're measuring mechanical properties, and then we're measuring gene expression, or using molecular inhibitors to inhibit specific signaling pathways. And that's generally our approach, so that we can understand not only how the forces are affected by certain perturbations, but how that affects the signaling pathways that control those forces. And we had a very lucky result, and that was that we saw that the ovarian cancer cells, which metastasize to the softer tissues, exert higher traction forces on softer environments. And those traction forces are also more polarized. And as others have shown, the breast cancer cells express higher traction forces on hard surfaces, and again, they're more polarized. And we measured these things for hundreds of individual cells, and then we wanted to find out whether those forces were correlated with any behaviors that we associated with more invasive cancer cells. And so we looked at several things, including motility, which we expect to be increased for more invasive cells, proliferation, and chemotherapeutic resistance. And so what we find is that when the cells exert higher force, regardless of whether it's on a stiff or a soft environment, they are also more proliferative, and so higher expression of BRDU, which is a proliferation marker. They remain viable when we treat them with drugs like carboplatin or Taxol, and they're more migratory, and so their velocity is higher, regardless of whether they're on a soft or stiff environment. And so we wanted to find out whether there were any intrinsic differences in these cells. And so we looked at gene expression, and we found that the cells that express higher forces on stiffer environments also express higher levels of these actin-myosin contractility genes. And so then we thought, if we manipulate this pathway, what will happen to these responses? And so we confirmed that the higher expression was also seen on cells grown on their preferred environment. And we saw higher expression, so these are 2 markers of non-muscle myosin 2, And we found higher expression on hard surface for breast cancer and a higher expression on soft first off. This is the pathway that controls actin, myosin contractility. I told you a little bit about this pathway earlier, but this is a diagram of that pathway, which shows in red and green some things that you can do to affect this pathway. LPA is a drug that's used to activate the signaling pathway to induce actin, myosin contractility, and Y27632, ML7, and blebbistatin are all inhibitors of this pathway that work at different points. And so we use these inhibitors and these activators to manipulate the signaling pathway to see if we can alter the response. And what we found is if we treat with inhibitors, we get responses that are independent of the mechanical environment. And I'm only showing you a small amount of the data. There's tons of measurements we made, but regardless of whether we're measuring force, spread area, velocity, chemoresistance, if we treat with Y27632 or MO7, the difference between soft and hard is abrogated for both cell types. And so why is this important? And so this is some data that shows the velocity fold change between soft and hard. And I mentioned that only invasive cells respond to stiffness of their environment by exerting higher forces and having these more invasive behaviors. But when we look at non-invasive cells, we get no change. And so if we take these more invasive cells and we treat with inhibitors of this actin myosin contractility gene, we end up with invasive cancer cells that begin to behave more like non-invasive cancer cells. And so I think this could be a way to kind of reverse some of these phenotypical characteristics that we're seeing for these more invasive cancer cells. And so I've already told you guys about this study a little bit, and I want to go back to it for just a second, and I want to tell you one more thing that's important about it. And that's that when we have a cell that's invasive, it responds to a change in the mechanics of their environment, so it's mechanosensing. But if we take a cell and we induce it to express something like snail constitutively, and so it has this more invasive phenotype, then it no longer responds to changes in the stiffness of its environment. But the higher force profiles, the change in intracellular mechanics, are always that way. And so we think that it's not just that these cells are responding mechanically, but they're undergoing some sort of EMT that leads to this mechanical response. And this is more on that. I showed you this slide already. And I want to give you one more example of how we've used this to look at changes in invasive cancer cells. And so one thing we did was we isolated Taxol-resistant clones of these invasive cancer cells. And then we did similar characterization of forces. And we find that just having cells that are Taxol resistant increases their force profile. And it alters their adhesion. And if we treat with something that affects the way these cells interact with their environment, like a focal adhesion kinase inhibitor, mentioned that focal adhesions are these force transducers between the intracellular environment and the extracellular matrix. And if we inhibit FAC, which is a protein that's present in that focal adhesion, then it reverses a lot of these things. And so now we can use these types of biophysical measurements to screen drugs to see if they can reverse these mechanical phenotypes that we measured. And so what I would like to do with this in the future is apply this tool that we've developed to look at different types of cells. I mentioned that we have already isolated and characterized Taxol-resistant cells, but we've also isolated carboplatin-resistant cells and cells that are more resistant to both Taxol and carboplatin, as well as other drugs. And so we have all of these different cells that we would like to be able to run through with these biophysical characterizations. And so to enable this, we need to be able to do this in a more high-throughput platform. And so traction force microscopy can be a very laborious process where you are measuring the motion of these particles in a gel for one experiment and then repeating that experiment 3 times for every cell type and then 8 per patient, and it just keeps growing. And so being able to adapt this to a 96-well plate and use a surface that is uniformly flat is one of the things that we plan to do in the future, to be able to do this in a more high-throughput way. If we have this device, then we can use it to look at different cell populations, so this will look at different types of cancer cells. We can also use it as a screen for different molecular inhibitors or therapeutic drugs, and it's not only applicable to cancer cells, but any kind of cell that you want to put in. Another type of tumor cell that I've been looking at is a non-malignant tumor cell. These are cells that are isolated from women that had hysterectomies, and they're from tumors that are called uterine fibroids, and these are very fibrotic tumors, and these tissues are stiff. And so I was really interested in how these fibrotic cells that grow in these really stiff environments grow when the myometrial cells aren't. And so normally myometrial cells grow much better than the fibroid cells in vitro, but if we apply a mechanical load, then this behavior is reversed. And so we've been looking at developing ways of kind of studying this more. And we've started to make some measurements. We've done some microarray studies to look at gene expression, and there's hundreds and maybe even thousands of genes that are being differentially regulated. We haven't sifted through all this data yet, but we know that there are things like TGF-beta signaling pathway that's heavily affected, ECM interactions, and bulbar regions. And these are things that are involved in all the mechanical measurements that I've mentioned. We also know that if we apply a mechanical load to fibroid cells, we undergo stiffening. But if we apply the same mechanical load to the myometrial cells that they form from, they don't undergo this stiffening. And so we think that there's something that is transformed within these cells that allows them to respond on an intracellular level to these kind of mechanical stresses. And these are some of the assays we've developed to analyze fibroid cells. This first one we used for cancer cells. We were interested in seeing how spheroids respond to stresses. And so we had done a lot of studies with this type of tool for looking at spheroids in culture or for monolayers that kind of mimic spheroids. And what we do is we grow cells in a monolayer, then we apply a gel to the surface, and then we have weights that we apply to the surface. And we can inject particles in these cells before we see them on the surfaces. And so we can do particle tracking, and then we can do all kinds of crazy stuff with it. And so it's a really neat experiment, and it's very simple. And when we apply the weight, we know that there's changes in mechanics. We can also use our polyacrylamide substrates, measure things like traction forces, or we can culture these as spheroids and see how they respond to this 3D environment. So the last part of my talk, and I know I don't have much time, so I'll just go over a few of the slides in this last, and then I'll talk about teaching. But this last part I think is really interesting because it shows how these things fit together. And so we've been interested in seeing how cancer cells respond to stem cells and how stem cells respond to cancer cells. And I've already mentioned how the stem cells are present within these tumor microenvironments and how they contribute to the pathology of these conditions. And so we have several different types of tools that we use to look at this. One is a very simple adhesion assay where we can grow cancer cells in a monolayer, and then we can take fluorescently labeled stem cells and allow them to attach. And then we apply force, which detaches the cells, and then we can measure the number of cells that remain attached. And then we can look at the expression of cell adhesion molecules that allow them to attach and find out whether specific cell adhesion molecules are involved in that attachment. And so this is a really great tool. We can apply it in this context, but it's really applicable to a lot of other situations as well. The other one is a more laborious process where we track cells in a 3D gel. And we can also embed particles within these gels, and the particles will be displaced as the cells are moving within the gel, so we can get like a pseudo-traction. These are collagen gels, which are not perfectly elastic, so it's not really a traction force, it's just a beam displacement, but it's very similar to the kind of measurement we make when we're making the traction stress measurement. And so this is our adhesion assay. Very simple. We have fluorescently labeled cells that we attach to a monolayer, we then remove them, and we make a measurement. These are images of our cells in blue. This is a DAPI stain, and it shows that the monolayer is continuous on the surface. And then the green are the stem cells that are attached to it. We did this with stem cells, and we also did this with fibroblasts. And the behavior in this area is the same. And so the stem cells or fibroblasts adhere similarly to these surfaces. And what we found in every case is that stromal cells are less adherent to non-invasive cancer cells, regardless of whether this is a prostate cancer cell line, an ovarian cancer cell line, a breast cancer cell line, a less resistant to chemotherapeutic cell line. If we have a less invasive cell line, then the stem cells or the fibroblasts will be less adherent to it. And so this was a pretty remarkable measurement that my student made, and it was very simple in the way that she designed the study. And so we looked at the expression of certain adhesion molecules that are involved in interactions, including integrins. But in this case, we found it wasn't the integrins, but it was the cadherins that were really affecting this process. And cadherin 11 is an integrin, is a cadherin that's expressed by stromal cells, but it's also expressed by some invasive cancer cells. And then cadherin 2, or N-cadherin, is one of the cadherins that's upregulated when cells undergo ENT. And so we found invasive cells express more of both of these cadherins than non-invasive cancer cells. And that if we block cadherin 11 on the stromal cells, that it prevents them from being able to adhere, regardless of whether the cancer cells they're adhering to express N-cadherin or cadherin 11. And so cadherin 11 can interact with N-cadherin or cadherin 11 on the cancer cells, which would allow them to attach. And in this last study that I want to just really briefly touch on, we did 3D tracking of these cells, and we looked at breast cancer cells moving alone. And so this is a breast cancer cell, and you can see that it kind of starts to escape its little location, but it doesn't translocate. But if we add a stem cell to the culture, it starts to move further. And if we look at this quantitatively, we see that if for the stem cell behavior, there's no change in migration of the actual stem cell. If it's a non-invasive cancer cell, there's no change in migration again. But if it's an invasive cancer cell, we get higher, and this is the directional velocity measurement, and if we use MSC conditioned media instead of adding an MSC, we mimic that. And so we also found that the pathways that regulated other processes on a mechanical level were important in this process as well. And we focused on several of them that are really important in three-dimensional migration, like matrix metalloproteinases, row-associated kinase, focal adhesion kinase, and so forth. And after looking at many soluble factors, we ended up back to our old friend TGF-beta, and we found that TGF-beta was actually being produced by MSCs, and that was inducing this directional motility response in the cancer cells. We showed that by mimicking the response by adding TGF-beta instead of MSCs or MSC-conditioned media, and then we also showed that by blocking TGF-beta in conditioned media from MSCs. And then we went back to the signaling pathways that were affected before, and we saw that if we added TGF-beta antibody, that also inhibited those as well. And so I've told you some stories that all involve mechanics, and they also involve biology. And a lot of what I do involves pretty complex biology and understanding of signaling pathways and how they affect cell function, especially in the context of cancer. But mechanics adds to that. And oftentimes it's the mechanics that really allows us to understand what's happening better than if we just measure gene or molecular expression. And so this is a summary of what I've talked about, but I don't want to cover that because I do want to mention teaching. And so since I've been at Georgia Tech, I've taught many semesters of the same classes, including heat and mass transfer. We use Welty, Hicks, and Wilson, momentum, mass, and heat transfer, and as a supplemental text in fluid mechanics, I use Fox and McDonald. In heat and mass transfer, as a supplemental text, I use Invermer and DeWitt. I mention these books because I know that these are more mechanical engineering version of these textbooks. Heat and mass transfer is something I've been teaching for a long time, so I really enjoy teaching heat and mass transfer. And fluid mechanics, I've taught a few times, and I really like fluids because it's directly related to what I do in my research, and so I'm always trying to expand my understanding. And that is something that you can do when you're teaching a class. And another thing that I teach is biomolecular engineering of the cell, which is a chemical engineering version of cellular engineering. This is a class that's attended by a lot of undergrads. I have high enrollment in my class, much higher than a lot of the other electives, and so I think the students have really responded well to what I've put together for this class. There are several courses that I would be interested in teaching in mechanical engineering. Of course, heat and mass transfer. I think it might take some time to adapt kind of the chemical engineering version to the mechanical engineering version, but that's something that I would love to do. And then biofluid mechanics. I think that increasing my knowledge of fluids is really, is something that I'm really interested in doing. I use fluids in my work, and I like teaching fluids, but it's something that you need to teach several times before you, I guess, are a master of it. And so I think that I'm still growing in that respect. There are also bioengineering classes that I would like to teach. I don't really know much about mechanical engineering 570, but if it's a bioengineering class, I think I could probably teach it. And cell and tissue engineering. I would like to see cell and tissue engineering in 2 classes, one that's focused on the biomolecular stuff and one that's focused on the tissue level stuff, and I would love to teach it as a 2-class series. There are some other classes that I would love to be able to develop. One is engineering applications in tumor biology, and this would be directly related to a lot of my work. But I would also like to start like Weinberg does. So Bob Weinberg is one of the people that first identified the genes that were mutated in cancer cells, and he has written several textbooks on cancer and what cancer is. And so it starts with this molecular definition of cancer, and if students don't understand cancer on a molecular level, I don't think they can understand the functional changes in cancer cells later. And so I think it needs to start with the molecular definition of cancer, and then how cancer affects the microenvironment. Oftentimes when people that don't know a lot about cancer think about cancer, they think of it as this group of cancer cells, but it's not just a group of cancer cells. It's a group of cancer cells that has recruited these host-derived cells that form the stroma, and without the stroma, the tumor is just going to sit there dormant. And so this stromal recruitment is absolutely essential. And then experimental techniques that are used to detect cancer or to measure mechanical changes in tumor cells. All of these things could be incorporated in this class. And then I've mentioned cell and tissue mechanics. This would cover cytoskeletal mechanics, techniques for measuring tissue stiffness, methods for modeling in the lab. And then this is my biomolecular engineering of the cell class. And so it starts with an introduction to cell biology, then more advanced molecular and cell biology techniques, and then it ends with special topics. And I break it into this kind of series of segments. This first one makes sure that students that don't have a strong background in biology have the fundamental understanding that they need to be successful in this class. And the second part is more advanced topics, things I've mentioned here, molecular cloning and genetic engineering. I think that's one of the things that the students like most about the class, is having that chance to really learn that formally. And then we talk a lot about receptor-ligand interactions, using Laufenberger and Bell models. And then we talk a lot about cell fate processes and how we can measure cell fate processes. And then the special topics every year increase because there's so many new topics that are being brought into biomedical engineering. This year, some of the new additions were the brain and the microbiome. The microbiome is a growing area in BME and something that I think we should all know something about. I have written a lot about my teaching philosophy in my package, and I think that this is, kind of a repeat of some of that. But I would like to try to foster an active learning environment in my classroom, so I do as much as I can to try to initiate that. In large classes, it's difficult, but I think you still need to incorporate tools for doing that. And so I've listed a couple things that I do specifically. In my core courses, I use written quizzes. Written quizzes that are simplified versions of what you might expect on an exam, but that are broken down into the essential steps. And the student works through those steps. Oftentimes they're very unsuccessful in their first attempt, but hopefully by the time they get to the exam, they will really understand this, and then, more exams can be better and easier for me to grade and to assign points that the students will be happy about. Elective courses, I think one thing that has really helped me is this journal presentation, but I use a very different model for journal presentations in the class. There was an article about the 10-minute journal article presentation, and I use that as a basis for how I have designed these topics. And I specify the topics, I have strict guidelines for article selection, and this process helps students to learn how to review the literature, and then their final project actually builds on that because they have to do a literature search, which is part of the project. And so overall, I think that by the time they finish this class, they really understand the literature a lot better than when they started out. Many of the students, when they come in, they think that a Google search is how we find a journal article. And as graduate students, I'm sure that you don't think that anymore, but maybe as an undergrad, that's not so uncommon. And so I'll stop here. This is kind of a summary of my research area, bioengineering, and my lab.
Thank you. We have time for questions.
Thanks a lot for the talk. I really enjoyed it. I was wondering physically how you measure your traction forces in the lab on the cells.
So we have a MATLAB code that we use to analyze the traction stresses. But what we are measuring basically is we are imaging the cells on the surface of the gel, and then we're detaching the cell. And so we use SDS, which is a very potent agent that basically detaches them from the surface rapidly. And then we take a picture of the gel again. And so the particles move back to their, since it's elastic, the particles return to their original rotation. And so we have these two images, and then we use a cross correlation function to determine the displacements. That gives us our strain field. And then we determine the traction stresses. And so there's the Green's function, which basically characterizes the elasticity of the gel based on the Young's moduli. And then there's also the parameters that are kind of in there. And then we have the displacement field. And so we relate that displacement field to the Green's function and to the stress. And so basically we back calculate the stress from this field.
Okay. And do you use typically just like a Young's modulus for your precision model, or does that?
Yeah, so we use the Young's modulus, which is a pretty good measurement for polyacrylamide. It wouldn't work for collagen because it's not an elastic material. But maybe linear behavior that we could over a certain range, so we may be able to use it in certain contexts, but it would be a stretch.
Have you looked at all about including some type of like nonlinear type of constituent formulation? Is that?
It's really difficult because the computation it takes the computer a long time to be able to analyze these for a few experiments. And so if we made the model more complicated, we would need more computational power to be able to run it, and probably need somebody that had a background in mathematics to be able to help me make sure that that wasn't incorrect. And really,
So I have a kind of related question in that I was wondering how you do the statistics on your traction force microscopy, because I think I saw you did statistics on it later, and then can you only do it in the 2D, or in your 3D models can you also do that kind of thing?
So we do traction force microscopy on hundreds of cells for each condition. So every measurement represents 3 experiments with multiple gels, and so it's a large amount of data before we're ready to report a measurement, and I think that's essential. And the statistics are built into the model. I don't know exactly what we're calculating in because I didn't generate all of the code, but we are using the code to generate our statistics as well. And let's see,
3D.
your question 3D. So in 3D we do beat displacements, and so I showed that picture of the
Oh,
gel.
that's right.
I'm gonna go back to it because so these round circles that are out of focus, those are beads that are embedded in the gel. And so as the cell moves, it kind of moves the bead, and so we can measure the displacements, and there have been some papers where they looked at the net displacement, and that is kind of the 3D pseudo measurement of traction. But it's not traction, it's just a bead displacement. But it's similar in that it gives us some idea of how much the gel around the cell is being deformed. But we don't know whether the gel is being deformed in the z-axis. We only know whether it's being formed in this direction. So we're assuming that it's an isotropic media when it might not be.
I like your idea of comparing the molecular and mechanical changes of the cells from a young population to an aged population. And presumably that would be similar in vitro work to what you've shown here. But I wonder how you control for things like the endocrine and paracrine changes that occur from young to aged, as well as things like changes in the matrix, such as the increased cross-linking of a lot of collagenous tissues that occur with aging.
So the increased cross-linking, we wouldn't be controlling for, but hopefully if we measure their behavior on different substrate elasticities, we may see something that might help us to understand whether or not there's some factors related to the citizen and the environment that are activating some of these processes. One thing I would like to look at is exosome secretion. And so we know that human MSCs, as they undergo aging, produce these vesicles, and these vesicles contain a lot of things that alter the environment. And so I think that might be something that happens there that might help us to look at the paracrine effects. And And so I would like to be able to isolate those exosomes and then give them to the cells from the younger donors and see whether that changes things. And so, you know, some of the media tricks that we play might help us to kind of parse some of this stuff out. But I mean, that's what we would basically be doing, because we are really simplifying a system. We're saying, okay, is the cell inherently different? And now if we add back in things that are present within the tissue environment, does that give us behavior that's more like the behavior that we're trying to study? And that's kind of the best we can do at this point. Maybe, you know, adding the exosome will definitely add another complexity to it.
Any other questions? Ben, you probably have a few more.
Internally.
I thought it was kind of curious that the cell, when it wanted to move, it had to switch from being viscous to elastic. Is that just because internally you need some mechanism for transmitting force throughout its interior?
So, when a cancer cell is undergoing EMT, it becomes softer, and when a stem cell wants to move, it becomes stiffer. And so, in the context of the stem cell, it might be that there's actin filaments that are moving, and that there's activation of myosin within the cell that is contributing in some ways. We think that it's because the cell is elongating, and when it elongates, the stress fibers are being squished together, and so the particles are just stuck within those filaments, and they don't have the ability to escape. And so, and there's also branching of the actin at the periphery of the cell, which would trap cells, particles that were near the periphery or in the cortex of the cell. And so everywhere within the cell, the structure becomes more confined. And so we think that it's a confinement issue. But, you know, we don't really know whether it's going to lead to a motor-related response. And so that's one of the things we need to do for that PDGF, TGF beta 1 story. So we haven't really followed up on that yet.
Okay, thanks again.